paper are red, blue, green, and yellow. Let's start with preparing the red paper to represent tDNA region, which contains tumor-causing genes. Next is the yellow paper for the origin of replication. This region ensures the plasmid can replicate within the bacterial cell, allowing it to be passed down and continue its function. Now for the blue paper for the virulence region. This part of the plasmid carries genes responsible for DNA transfer. And lastly, the green for the open catabolism. This piece enables the bacterium to use plant-produced nutrients, helping the bacteria survive and thrive once it's inside the plant. We paste all together the regions and label it. The white paper represents the border sequence. These border sequences guide the plasmid's interaction with the plant's genome, marking the beginning and end of the tDNA region. In the next step, we'll assemble these regions into cohesive structure, but first we need to finish label each component.
So hello everyone. So now I'm going to discuss these 4 following questions. So number 1, which region would be transferred to the cell? So of course, tDNA region of the plasmids. Number 2, what happens if the B region is damaged? Of course, the plasmid loses its ability to transfer to the plant cells. Number 3, why are the border sequence critical? It is because it represents a start and end of tDNA. Without this, it will not be properly cut and transferred. And for the last question, how might scientists modify this D plasmid for beneficial purposes? Of course, scientists modify D plasmids by removing these harmful tumor-causing genes and inserting useful genes. This allows safer transfer of useful genes without causing any diseases. Thank you.
The large container represents the plant cell, and the small one is the bacterial cell. Next, place 10 beads of one color inside the plant cell container. These beads symbolize the plant chromosomes, in the bacterial cell container, place the T plasmid model. This plasmid carries the genes that will interact with the plant chromosomes. The transformation begins as the bacterium approaches the plant, it detects cues from the wounded plant tissue. This triggers the activation of virulence. Blue beads represent the Ver-D proteins responsible for the DNA, while light green beads stand for Ver-B proteins. Now the tDNA region is prepared. It is carefully excised from the plasmid, leaving behind only what needs to be transferred into the plant. To protect the DNA and guide its destination, a blue bead representing Ver-D2 protein is attached, forming what's called the tDNA complex. This complex is now ready for its journey. With the tDNA complex assembled, it's time for transfer. The bacterial cells lines up with the plant cell, and the DNA begins its passage. Using small connectors, we simulate this integration, joining foreign DNA with the host's own genetic material. The plant is no longer the same. The transformation has begun.